to our first Science Ask Me Anything. Cerebrum DAO, we're here with our science team, Dr. Maryna Polyakova and Dr. Peter Groenen. And without further ado, I'll leave it up to you guys. Enjoy the session.
Thank you, Yatan, and welcome everybody. Today we're opening the science call for you, and we're really happy to hear any feedback, any questions from you, and hopefully we will be able to answer them. So we would like to talk with you about how Cerebrum DAO approaches the translational gap, and how we define our topics, our focus, and how we approach the projects. We can go next. I guess for everybody in the field of neurodegeneration, it's not a secret that Alzheimer's disease is a great burden to our healthcare system. It seems, by the way, that the incidence is decreasing in the developed countries, but nevertheless, with the aging population, the pressure on healthcare costs is still enormous, and it's going to increase over the time. This is one of the reasons why Cerebrum DAO exists. And if we go to the next slide, you will see that we have settled us on a great vision, and we really would love to see the world with only healthy brains. We know that it is a long path. Nevertheless, we believe that our organization will exist long enough, and our aim is to contribute to solving those diseases. So our mission is to identify the original translatable research and help researchers to streamline it and accelerate the path to capital and, of course, to the clinic. We can go to the next slide. So today we're going to give you a little bit of the introduction. We know that we're followed by a variety of audience, so forgive us if you already know some of the information that comes in the beginning. We will talk about some of the pathological mechanisms which we think are currently not yet well translated and definitely are not in the clinic. Then we will talk about the innovation in the space and how our portfolio fits and brings new aspects to the market. And then we focus on the future steps, and we're happy to give answers to any of the questions that may appear. And on the next slide, we start with the Alzheimer's drug discovery pipeline. I think this article is also well known to anybody who is in the field. And as you see, a number of companies are working on multiple targets and are currently in the different phases of drug discovery and development. And of course, if you could see on the right side of the slide, the majority of projects are still, especially those that are in the later stages, they are focusing on the aggregated proteins, amyloid and tau, and some of them have reached the market recently. Nevertheless, everybody knows that the clinical, let's say, relevance is still suboptimal of those drugs that have currently reached the market. And what is very important is that the side effect profile is still very difficult, let's say, and only a minority of patients can actually be prescribed those drugs. So we are talking about the numbers of 10% or even less, which tells us that even with the current successes, there is still a lot of work, even in the field of Alzheimer's, and I'm not even talking now about other neurodegenerative diseases which are more rare. So we started our organization already over 2 years ago, 3 years ago. And in the very beginning, we were thinking, how can we approach it differently? Because one of the goals of our team was that we actually increase the number of hypotheses. And the way we decided to start is to look at the hallmarks of neurodegeneration and partially also on the hallmarks of aging. And we wanted to find the intersection of what actually we know about Alzheimer's disease, neurodegenerative diseases, and what are the processes which are affected in aging overall. Also, we were interested to address those topics that are present across diseases and not on the Alzheimer's specific. Although, of course, Alzheimer's was our primarily topic in the beginning. So as we all know, currently it's no longer a secret that the protein aggregation hypothesis does not solve the whole problem. Yes, we know that a number of proteins aggregate in different neurodegenerative diseases, but we also know very well now that some processes like neuroinflammation, like synaptic loss, aberrant proteostasis, and mitochondrial dysfunction, for example, they are present early on in the disease. And although it is a matter of discussion, some people say that some of them are really crucial to the development of pathology and to actual neuronal death. On the next slide, we also summarize a number of pathological changes that occur in the neurodegenerative diseases. So again, proteostasis failure, mitochondrial dysfunction, oxidative stress and reactive oxygen species, neuroinflammation, synaptic dysfunction, autophagy failures, and lipid dysregulations. Those are the topics that we were looking at. And today we will focus more on a couple of them, which we decided to explore wider. And the first one, of course, is the neuroinflammation. We know that a large number of groups are actually exploring the topic, and there are even certain clinical candidates already. Nevertheless, it seems that the progress could still be sped up. And we indeed found one project which we decided to support and to help bring further. So as you may know that neuroinflammation, glial activation are observed in a number of diseases. And as of now, we already think that it is, so neuroinflammation is a part of the vicious cycle. So in the beginning, glial cells are responding to the protein aggregates, trying to rescue the normal brain function, but later on they become hyperactivated themselves, and they contribute then to proteostasis failure. We also find them with the synaptic loss. We also find neuroinflammation at the blood-brain barrier areas, which become disrupted. And we also can observe oxidative stress in the neuroinflammation areas. So very often it's a vicious cycle, and it's really difficult to find a single starting point. Nevertheless, Peter will talk later about one of the hypotheses that we are helping to develop. Here's the next slide. Yes, we're going to the current understanding. And yeah, so one of the topics, as I mentioned, is the blood-brain barrier dysfunction. These changes that we see in the neurodegenerative diseases, they are very much related to the inflammation, and basically As you all know, that the blood-brain barrier is a very specialized structure in the human brain. On one hand, it is connected to the bloodstream, and basically it would allow us to transfer some of the drugs, for example, to the brain tissue. On the other hand, it's very selective, and is trying to is aiming to protect neurons via the selectivity. So we know that it allows only small molecules or oxygen to pass by the passive transport, and very specialized mechanisms exist to transfer glucose, and some immune cells can also pass it. So we know quite well that any inflammatory disease or any inflammatory state will open the blood-brain barrier for a while. And thus chronic inflammation and chronic protein aggregation, they are directly related to increased permeability. Therefore, blood-brain barrier dysfunction was one of the topics that we were actively exploring. We could go to the next slide, I guess. Yeah. Partially, I have also mentioned some of this information. Yeah, so over 60% of patients show blood-brain barrier dysfunction, and very often it is also coinciding with the amyloid pathology and the parasite loss. I can only say probably that in the course of our Cerebrum DAO activities, we looked actively into the research groups working with the blood-brain barrier dysfunction. And it turned out to be not the simplest topic for translation. Yeah, maybe we should discuss it separately in a longer session. We could go to the next slide. One of the recent topics that became finally, let's say, well discussed in the scientific community is the brain lipidome. Therefore, from the last year, we also started looking into the research on the lipids of the brain. So, brain is a very fatty organ. Over the 60% of brain dry weight constitute lipids. We also know that the enormous number of lipids have been already identified in the brain, over 600 species, and 30% of them are unique to the central nervous system. Various lipids have been explored so far or researched, such as phospholipids, sphingolipids, cholesterol pathways, and also omega polyunsaturated lipids. And at the current stage, it seems that we start to understand some of the mechanisms. We start to see some of the targets, but in terms of the progress for drug discovery, this topic is very, very new, and we are happy that we could identify a number of very interesting and promising projects. One of the first topics that we explored in the series of Cerebrum DAO research sourcing was the mitochondrial dysfunction. And although not many drugs have been advancing in this space, mitochondrial dysfunction is essential to the neurodegenerative diseases. As you all know, that although brain is less than 2 kilograms, has less than 2 kilograms of weight as compared to our whole body, it consumes an enormous number of energy, so 20%. It is well known that we have over 2,000 mitochondria per neuron, and over 60% of neuronal ATP is used for the natrium potassium ATPase. Yeah, so mitochondria are not only energy engines of the cells, although of course they are one of the main roles is producing the ATP. They also do participate in the calcium buffering, in the reactive species regulation, and in the quality control of the cell. Therefore, we are very much interested in this topic, and Peter will also present one of the projects which is already advancing the mitochondrial dysfunction field for the neurodegenerative diseases. So mitochondrial species, mitochondrial DNA and reactive oxygen species are closely related to the neuroinflammation in the brain, and we see that the mitochondrial stress markers are present at the locations of the accumulation of amyloid beta, tau, and tau. When those occur, then mitochondria become damaged and they release their DNAs into the cytosol. Yes, and I guess a number of targets could be explored in this area, which is also which will be further also discussed by Peter. Yes, and with this, I want to switch our topic more into the everyday work of Cerebrum DAO, which is searching for the drug discovery projects originating from the academia. And I will give the word to Peter further, as he is our main expert in this field.
Thank you, Maryna, and welcome everyone. So I will take you through some of the projects that we're working on. And the idea is that this session we have today is kind of a general introduction. We've been asked to go deeper into the science, and we will have more sessions to come where each of the topics that I briefly touch upon will be be discussed in at more length, and also be given more details about what we're doing, why we're doing it, and the deeper science behind it. But before I go there, I mean, this session is also about what we're doing to accelerate drug discovery and development, and to eventually, as Maryna was saying in the beginning, touch upon novel targets or novel mechanisms of actions. And if you're looking at the overall progress, specifically in neurodegeneration or neuroinflammation over the last, let's say, 60 years, there have been some significant findings, significant treatment options, where either we have disease modifiers or at least we are able to treat the disease. But if you realize this perspective that you're seeing here for the major neurological disorders, you have to agree that progress has been limited, and as Maryna was already pointing out, the current treatments for Alzheimer's disease, in the sense of disease modifying, not treatment of symptoms, but disease modifying, are considered to be very limited with side effects. And it is argued even by recent publications that they actually do not work at all. The question is, are we in the right population? Are we at the right time of treatment, etc., etc.? But if you're looking at this picture, you realize that progress has been rather limited, though there are some significant treatments nevertheless. And if you then look in more detail over the last 10 years, what kind of innovation we've had, there has been innovation. But again, mainly in other neurological disorders and very limited in Alzheimer's disease. So I'm going to take you through some of the projects that we're working on, and I'm not going to go too deep. And I'm first going to touch upon a project which we recently started, where our interest was raised by several discussions we've had with experts and looking into this matter, because galectin-3 has been emerging as a very interesting target, not just in inflammation, also in oncology, because it seems to be involved in a lot of different processes. And if you're looking at the characteristics of a protein, as you mentioned here, it's a very typical protein. It's involved in a number of processes, and it had been linked to a variety of neurological disorders and very tightly linked also to inflammation. So what has been suggested now over the last, I would say, 5 to 10 years is the specific involvement in brain disorders and specifically neuroinflammation. So galectin-3 drives the microglial neuroinflammation. It's elevated in Alzheimer's disease brains, and you can also see in several knockout mice that there's a clear correlation with regard to the AB burden and galectin-3 increase. And there's a number of processes that are being involved. Gal3 binds glycosylated TREM2, and TREM2 has of course already been mentioned quite a lot in the last 10 to 15 years with its involvement in Alzheimer's disease. So here's the connection with existing biology that we already know for quite a while. Maryna already touched upon this NLRP3 amplification, which is also clearly linked to the inflammasome. So there is quite an interest in galectin-3, but nobody has so far shown any particular effects in neurological disorders other than maybe in animal models. So we were interested in seeing if this was a project that we might be able to further accelerate and have a different approach than some of the other projects. Here, in principle, you see summarized by one of the major publications over the last years with regard to clear evidence in Alzheimer's disease brains of galectin-3 increase and galectin-3 with regard to the A-beta burden. And as we were evaluating this project, we came across a project which is in principle the route that we are taking more often in our different projects, which is starting with the concept of the protein, doing a very fast in silico screen and focus on some of the characteristics that would allow us to do a very quick testing. This group presented a study where they found a molecule by an in silico screen, which also was blood-brain permeable, so allowed actually to do a rather simple study to see if galectin-3 inhibition would actually alter the pathology in this particular mouse model. And as you can see here, it's not a very potent molecule, but nevertheless, over a 30-day treatment on the right hand, you can see that it had a significant effect in one of the major animal models for Alzheimer's disease, the 5x FAD. Now, this is one of the examples where we pick up on the science that is emerging, which is clearly not what the majority is following, and we are now setting up a project to replicate this study and to further understand with a number of parameters if what this study has been shown is actually solid enough. And we have lined out a number of ideas to follow up after we replicate the findings of this study. So this is a very promising novel approach, which is very early. It's in the discovery stage, but it has all the hallmarks of becoming a very promising drug discovery project. Another one, again, where Maryna already pointed out, where our interest goes is the mitochondrium. There is a clear involvement of mitochondria in aging. As we are aging in different cells and different organs, the effects of the degradation of the mitochondria has an effect on the household, the energy household, and it shows itself then in aspects like inflammation. That's why we often speak of inflammaging, because as we age, there is a number of processes which seem to kind of have a result of wear and tear and derail the normal cellular function, and therefore also show itself in pathology. And one of the idea was that people have been looking a lot at this process that is called mitochondrial fission, but blocking the DRP1 normally gives a lot of side effects, because the fission process of mitochondria is also a natural process that allows you to get rid of faulty mitochondria, and in principle also cleans up cells which are really, let's say, in a bad shape. So one of the researchers that we're working with, Lou Rios, already 10 years ago, was working on the interaction between DRP1 and FIS1, which is in a natural way regulated by a protein called P110. And this protein P110, sorry, this one slide too fast. This natural protein P110 was already screened for a small molecule, SC9, and this SC9 was already tested successfully in a number of different assays and also in vivo models, and showed actually a biological effect. Downside was that it was not a very potent molecule, and it was also not passing the blood-brain barrier. So we set off with a project where we screened in silico for over 5 billion structures which could be synthesized. And that was actually the previous slide. We were already successful in listing approximately 1,000 selected top hits, which we are now using as an enriched set to do in a primary screen, where we look at reactive oxygen species and see how in this assay it can be blocked. So fission can be blocked by a small molecule. So this is now currently ongoing, and this is one of the topics which we want to present to you in the future with more detail, more background, and hopefully also, if we do that in a couple of months from now, with more details of where we're going with in vitro assays to further prove that what we are seeing is actually a valuable route for new drugs. Another project which we already have presented more often in our town halls is, again, when we age, a lot of mechanisms are starting to show wear and tear, and one of the mechanisms that is clearly affected also about within aging is the ribosomal translation into proteins. The older we get, the more older the cells get, the worse the control mechanisms get. So we get more and more proteins which are faulty, which fold in an inappropriate way and then start aggregating. So again, protein aggregates again are one of the hallmarks of neurodegeneration, and we are very interested with a collaboration that we have at the University of Basel to further understand what are these mechanisms that lead to this infidelity in protein translation, and how could we actually improve this by blocking certain processes, or maybe, if we find out, by stimulating. And here again, the approach was more starting from a unique assay where we introduce actually an infidelity in the translation. So we create aggregates, and we're trying to find a way by stopping this infidelity of protein translation and seeing what the effect is. So we screened, or the researchers in University of Basel screened a small library with known compounds, and we're looking at the effects, and they identified actually 2 compounds that were able to improve the fidelity of the protein translation, so correct actually the introduction that we did in an artificial way. And they showed that there were 2 known drugs that are marketed, that are being used for various indications, for which we have at least now shown that they're able to correct the translational aspects again. So in this case, you reduce the number of aggregates that are being created. So these are 3 examples, and Maryna also mentioned that we have been looking at the blood-brain barrier as a topic for drug discovery, and we have talked to several early initiatives. And together with the examples that we just showed you on drug discovery projects we're working on, we really want to show you that we are taking directions which are currently picking up, are not mainstream yet, are clearly good starting points for drug discovery, and allow also for, let's say, an accelerated process. So what are our planned actions for the 3 projects that we just showed you? So for fission bio, that's the mitochondrial fission process, which we try to inhibit very selectively, not the overall fission process, but specifically this through mimicking the P110 function. So we're currently confirming the hits. So I just showed you a snapshot of a true primary screen that we did. It's a cellular assay. And we are looking at the hits that, if we can confirm them, and looking at the structural groups, how they cluster. We're trying to confirm them also by in silico analysis and by binding assays, and then see if we can further expand these groups and understand also the dose response, and looking at least a first relatively simple in vitro assay that can tell something about the function of inhibiting this process. Together also if they're safe or not. Transfidelity, we have identified the two pathways by these commonly used drugs. That's a process we see more often, repurposing. And what we really would like to do now is understand these pathways, and eventually also, and the benefit of generating new intellectual property is identify new targets. So I can tell you already that we have identified a number of potential targets, and the project is now on its way to further validate these targets and start a drug discovery process. Galectin-3, we are now starting a full replication study for the molecule that I just showed you by a group in China. And it will add a few other biomarkers to make sure that next to replication we get a further insight to how this molecule really inhibits galectin-3 and improves the pathology in this mouse model. Here again, we are going beyond just this model, but that's the next steps after we confirm that this direction and this study actually makes sense. This already mentioned to you. This already mentioned to you as well. So I just wanted to kind of finish before we start a Q&A. I'm just looking at the time where we are. I think we're doing good. Next to picking targets which are, let's say, not mainstream, which of course bear a lot of risk, because if you follow validated targets which already have a clinical validation, you're of course on a much safer route. But then again, in this space, as you saw, the innovation has been quite limited. So there is a good reason to step outside of the current mainstream drug targets and look for alternative mechanisms. The route there to a clinical successful treatment, of course, is much longer. But it is clearly something that we as an organization believe gives us a better grip on treating these diseases. So the identification of drugs, can be done in a very traditional way. And as we're speaking about AI these days and how we can involve AI, not just by the processes, but also about the original identification of targets or the identification of novel molecules, as we were talking about in the fission project, where we screened 5 billion compounds very efficiently, very fast, in principle that cheap as compared to the more traditional approaches where you do cellular in vitro screens of 1 to 2 million compounds. So in this process we want to accelerate and make it more cost effective, so to speak, democratize the early drug discovery. And then as you move forward by testing different concepts, we know that we can automate the processes to make it more affordable, but also to test more things in parallel. And we think that these combinations are leading us quicker to novel findings and therefore intellectual property that we can protect And protection of intellectual property is, of course, one of the key aspects of obtaining further funding that will then eventually lead us to smart clinical research. There's another aspect, as we have one study going, which we will start, which is actually about to start, where it's going to be a completely decentralized clinical trial, where we nevertheless are making sure that the setup, the design of the study is very robust and very thorough with regard to the clinical symptoms. But it's going to be better to monitor, faster in the sense of getting to your data, and also not unimportantly going to be more affordable, so to speak, cheaper to do a clinical trial. So all these facts together will allow us to do drug discovery and drug development more efficiently and, so to speak, fail faster and fail cheaper if we're talking about failing. So this is in a nutshell something we have already dealt with in our town halls in how we work, and I'm not going to go into detail too much. I think we wanted to stay with the science today and allow you also to ask questions to us with regard to the current projects that you've seen, the projects that are emerging, and also if you have ideas. We are of course very open to collaborate, because that's one of the sources. We go out, we look for original research, we evaluate this research. We have a very thorough due diligence process where we involve top scientists throughout the world that eventually help us to select often very risky but very good scientific projects that are potential, that are offering potential to create novel IP. So I want to finish. There's nothing without a great team, and we are small. And we're very lean in the setup and very efficient in evaluation of projects and making decisions on putting in front of the community if they find it interesting to invest in, but also to participate in. So this is an invitation not just to fund, but also to participate, because we're looking for brains to help brains. I think this is the last slide. I think we did a pretty good job on time, at least now, and this gives us plenty of time to interact with you. I want to thank you for listening and open up the Q&A.
Thank you, Peter. There are a couple of questions coming up, and I guess I will actually ask you to answer some of them. So one of the questions is asking whether we prefer any specific modality. Are we interested in biologics? Are we funding small molecules? Maybe you could share your view on this.
Yeah, that's a great question. And I will try to answer it concise. So for a long time in various therapeutic areas, there has been a shift from small molecules to biologics for various reasons. Targets could be addressed more specific with antibodies. Several mechanisms were not addressable by small molecules. For instance, protein-protein interactions have been suffering a lot from having, good small molecules to interact. We're finding ourselves at kind of the pendulum swinging back. So small molecules are still interesting, and we see that the chemistry is improved. We see novel targets which were, perceived undruggable to be druggable, also with small molecules. And small molecules have a number of advantages. They're slightly easier in the early processes to work with. They're also cheaper in a setup, and in a sense, also from a therapeutic point of view, they're still offering the better class of molecules as compared to biologic. Biologic have a number of limitations, you need to have injections, although there are some improvements as well. So we still prefer small molecules over biologics for various reasons: complexity in drug discovery, costs of therapies for future use. But if a good project comes across which allows us also there to open up a novel route, I don't think we would close it. Neither would we do it for any of the other modalities that are now either evolving gene therapies, working with nucleotides. There are there are several other modalities now that are emerging, but I still think that, for the reasons I just mentioned, that small molecules are still going to be our main class of molecules we're going to invest in.
Thank you, Peter. One more question, I think I can take this one. How do we find our projects, and how is it possible to work with us? We have multiple ways to screen for projects, and I think over time, as our team has been developing, as our network has been growing, several pathways have already taken place. So the main way to find the projects is still sourcing. So we typically identify one or two areas which we're interested in. And Michele is our business development partner, and he is also sourcing actively for research in those certain areas of interest. We also have the application form on our website, so feel free to apply with your projects there. And if you have any questions, then you can always go on our Discord, interact with us there, or write us a personal message. With regard of working with our team, we typically first have an introductory call, and we identify your interests and possible pathways in which we could work together. There could be a number, and I think that, let's say specifically for our translational neuroscience, we really love to increase our network, recruit people to become our reviewers, and we consult the network members based on the project needs. So we regularly invite people to review projects. So feel free to contact us, drop us a line on Discord, drop us a personal message, and we will get in touch. All right. One more question is a tricky one, so I will give it to Peter. Do you use or do you plan to use animal models? And what is your view on the current current goal? Wait a second, to reduce animal testing?
Great question, and has my personal interest. I could talk an hour or longer, but I will try to, again, I try to answer this very concisely. Yes, at this stage, as I presented already one of the projects, we will still be using animal models. And we are very well aware of the limitations in animal models with regard to translation. We are also very much aware of the ethical considerations with regard to the experimentation due with animal models. Unfortunately, there are at the moment no real shortcuts. We do evaluate our models very carefully. As you know, statistics, they say all models are wrong and some are useful. This is the same for animal model usage in drug discovery. All models are wrong, but some are useful. And that's also in the example that I gave you, that we're going to replicate the study of galectin-3. We will put a lot of effort in with regard to thinking of alternative ways to get to the point that we understand if this has clinical relevance or not. Because that's the whole point about what we're trying to do. There's no benefit in treating mice or rats. Most of the time the disease that they represent has nothing to do with the human disease, and it's very artificial in a way that we're trying to understand. The Animal Usage Reduction Act, I don't know exactly what the name is, but let's say the global efforts to reduce the usage of animal models in biomedical research, of course, are very important. Health regulatory bodies like the FDA have clearly give out guidances in which they encourage sponsors, meaning companies or research institutions to reduce their animal models or actually come with better alternatives, because I think that's the whole thing. It's not just about reduction. You have to come up with better models, better alternatives for animal models. So there are several things that are happening now at the moment, and I think not wanting to kind of use the buzzword AI all along in our presentation, but AI, of course, as it presents itself, offers a great opportunity to get a much better mathematical model, a predictive model, an in silico model for certain effects. So hypothetically, you can agree if we would understand the whole mechanism dynamically of a cell and an organ and an organism, you could just interfere in the process and study over time what happens, and this would kind of predict if it would have an effect in the disease or not. Unfortunately, our understanding of disease and also of treatment is very poor, so we are limited. But several routes now are opening, like organoids, kind of culturing smaller organs on a dish, or allowing cells to be present on what we call organ on a chip, which allows you to screen many effects at the same time. So there, at the moment, you can see in this space of automation, but also in measuring, that there's many new ways that are emerging that allow us to avoid the use of animal models. And this is clearly also what we're looking at. We do sometimes get project presentations where the researchers really have taken a new route. And you can imagine that's sometimes difficult to evaluate, because you're used to kind of look at animal models, and you're kind of used to kind of see that what you see in animal models must have some kind of an effect, rather than looking at a chip or a chip readout. But we're seriously looking at it. But to give you a direct answer on your question, yes, unfortunately, we still are using animal models, but we're seriously looking at all places and talking to all partners that we know in our network to kind of reduce it and find better alternatives.
Thank you, Peter. There is one more question which we frequently hear from researchers, so I will take this one. What if we don't have drug discovery expertise in our lab? Can you help with that? That's a typical one, and I guess it's also quite clear to everyone that all of us have our own expertise. So, for example, even I'm more focused on the clinical aspect of Alzheimer's disease and other dementias. Peter is in a drug discovery. And very often we hear presentations from groups which are running basically academic biological research. They have a target, they have identified some mechanisms, and we do help them with the next stages. As Peter has mentioned already, we have partners which are helping with the in silico screening. We are currently increasing the network of the contract research organizations, which can help us with chemical synthesis or with some assays which are not available, for example, in some lab. I guess the ideal scenario is that researchers have some expertise. Let's say they can do the lab work, and we help them with the in silico screening, or we involve those CROs in further aspects of the work. Yeah, so we can help, and I guess the real advantage of Cerebrum is actually the network that we have, the variety of experts who can help with each of the very specific topics. And I have to say, it already saved us some money, involving right people at the right time. All right. There is one more question which we sometimes hear, and this one I will give to Peter. Are you interested only in Alzheimer's disease, or you're also looking in other diseases?
Great question. And thank you. I mean, the questions so far have been very good and very to the point. Yes, the short answer is yes. As you saw by Maryna's presentation and also looking at our current projects, they lend itself for other diseases than just Alzheimer's diseases. And there are some hallmarks about, aging and, neurodegeneration. And I think our current understanding of classifying diseases in a very strict way is also slowly disappearing. We do understand that there is no such thing as just AD. AD is many different diseases, and we see also a lot of comorbidities. Comorbidities means there's other underlying diseases. So it's also much better, and so it's actually a much more of a novel approach to leave out the very strict disease starting point and study diseases in a more holistic way, because we start seeing very many overlapping processes and the underlying mechanisms are very much resembling diseases like Parkinson's disease and Alzheimer's disease and others. So that's how we came also to more unifying mechanisms across these different diseases. So yes, we've been looking at Alzheimer's disease, Parkinson's disease, ALS, motor neuron diseases. We have been looking at epilepsy, and we have a focus on Alzheimer's disease for various reasons. Also because of our founder, who has a very strong relationship with Alzheimer's disease and Down syndrome. But I think it would be wrong to kind of only focus on Alzheimer's disease. And a lot of drugs are found by serendipity. By studying one disease with one molecule, you start understanding that actually it may not work in AD, but it may work in PD, some Parkinson's disease. So I think one of our strategy is to, certainly in the drug discovery process, to leave the hypothesis of where this now exactly will help as a treatment, to leave that a little bit on the side and to focus, as we say, where does the biology take us? Where does the molecule lead us? And where would it fit best in a sense of treatment in a clinical setting?
Yeah, I think that one thing that I could add to this answer, thank you, Peter. Yeah, your experience is always great. One of the diseases where we're looking in particular is indeed Down syndrome, because people who have touched it know that patients with Down syndrome have very high risk of Alzheimer's disease. So in a way, it is another genetic type of Alzheimer's disease, but also patients with this trisomy have multiple other diseases. So one of the initiatives that we are currently developing is a platform company focusing specifically on Down syndrome, and we're currently identifying the best path forward with the exact targets. All right, I think that we are at the end of our time, but there is one question which is difficult to leave because we both have opinion on it, and indeed we have had a couple of presentations on biomarkers. So the question is whether we are also interested in biomarkers for neurodegenerative diseases or only on drug discovery. Biomarkers is your space for sure, Peter.
Yeah, thanks. And yeah, this is a great question again. Sorry, it's very good. I love the question. Yes, we are also interested in biomarkers, for sure. And I believe that without getting a better understanding of disease diagnosis, but also understanding pathology and eventually the interaction with drugs, we will not progress very much. I believe this is one of the reasons why there is so much limited innovation, specifically in brain disorders, because it is so difficult to have a readout other than what you can see from the outside. So yes, biomarkers, if it's now plasma biomarkers, if it's now digital biomarkers, meaning measures of EEG or other means in which we get access what's going on in the brain, are of great interest. There is a bit of a downside to biomarkers slash diagnostics. First of all, it's not easier or more difficult than drug discovery, but it has its own particular challenges of doing thorough biomarker research. Secondly, from a funding perspective, certainly with regard to commercializing, things become a little bit more tricky. Unfortunately, the interest in biomarkers and in funding biomarkers for commercial purposes is much more limited as the interest there is for therapeutics. But we're trying to kind of work against this kind of paradigm because, again, to confirm, I think once we understand the pathology better by better biomarkers, we also be able to find drugs easier and find better drugs. So it is quite tightly bound together, and you could argue chicken and the egg, what do we do first? But I think we should invest more on the biomarker side than trying endlessly to find new therapeutics without really understanding what we're looking at, because we don't have such a good insight in the brain and how the brain evolves in the disease, but also in a treatment.
Thank you, Peter. From the clinical perspective, of course, we are coming closer and closer to the stage when we will be diagnosing not only specific clinical syndrome, as we mostly do now, but also really the biological subtype. And here biomarkers play a crucial role. Thank you so much for your questions. Those were quite a lot and really good ones. Yeah, I guess we were very interested to prepare this presentation for you, although it was still quite, let's say, superficial on most of the aspects. Nevertheless, we are aiming to create several more town halls where we actually go deeper into the projects that we are having, and also we will touch upon the neurotechnology, we will touch upon the replacement, which is, let's say, a hot topic in certain circles. And we are very much interested to hear your feedback. So contact us on Discord, contact us on Twitter, and if you have any questions, if you have any interest, we are happy to speak to you. And see you next time, I would say.
Thank you for listening. Thank you for the questions.
Thank you.